that would be cool if there were.
Yeah.
So this is all temporal bone. This is inner ear, not external or outer ear, not middle ear, strictly inner or internal ear. What you're seeing is an anterior view here. Now you see this portion of the inner ear that looks like a snail shell?
Yes.
It only looks like a snail shell in an anterior view. So try to do this, and you see it posterior view. That lets you guys see the anterior view now. Posterior view, it doesn't look like a snail shell.
Right.
All right? So, point being, this snail shell-like structure is called the cochlea. And the cochlea is the medial end of the inner ear. It's the deepest part. It's closest to the midline. So this is the medial part of the inner ear. So the midline would be here. Median line would be here. So which inner ear is it?
That is the right.
Anterior view.
Anterior.
Midline here.
Okay. Left.
Yes, this is the left. So now, yeah, so this would be slides right off. So it would be like so.
Got it.
All right?
Yep.
So the cochlea is always closest to the midline. All right, so, and on the lateral end of the inner ear, we've got 3 semicircular canals. 1, 2, 3. They're named. So this would be the left anterior semicircular canal, the left posterior semicircular canal, and the left lateral, lying on its side, lateral meaning to the side, left lateral semicircular canal. Again, all temporal bone, and within each semicircular canal, there is a tube, starts this gray tube here, called a semicircular duct. So this is specifically the left anterior semicircular duct within the left anterior semicircular canal. And I added a couple of types of fluid associated with the inner ear, endolymph and perilymph. See the pink fluid around the semicircular duct? You can see that pink fluid in here around the semicircular duct. That's perilymph, so peri meaning surrounding. Endolymph meaning inside, endo meaning inside. Endolymph would be the fluid inside the semicircular duct. So the interior of this duct, this membrane tube, contains endolymph, and then between the tube and the bone, around that tube, all the way around that bone, would be the perilymph. And in terms of the ion composition, they are different. They're both derived from blood plasma, but they're different results. So when they're made from blood plasma, but the actual ion concentrations are different in endo versus peri. So we see this posterior view. You can see the left posterior semicircular canal. The bone's partly cut away to reveal the semicircular duct within it, and you can see the pink perilymph in there as well, around the duct. Okay, so back to our anterior view. What do we have running between the more medial cochlea and the more lateral semicircular canals? We have what's called the vestibule. All of this, from there all the way to there. It's all vestibule right there. The middle portion of the inner ear would be the vestibule. And lifting this off, you can see there are 2 membranes in the vestibule. An oval window and a round window. There. You can see 2 membranes. The larger oval-shaped oval window, the smaller round window. 2 membranes in the vestibule there. So that's not bone there. So that's membrane holding back fluid. And what are the 2 types of fluid again? Endo and peri. So we have both types of fluid throughout the inner ear. Okay? What else? Flip this around to a posterior view, and you can see 2 branches of a cranial nerve, specifically cranial nerve 8, the vestibulocochlear nerve. So coming directly from the rear of the cochlea, we've got the cochlear branch that I'm gripping. Coming from the vestibule, we have the vestibular branch, and here they are about to converge. And so when the vestibular branch converges upon the cochlear branch, they actually do merge and form cranial nerve number 8, the vestibulocochlear nerve. In terms of function, the sensory impulses that travel along the vestibular branch, therefore the sense of equilibrium. The sensory impulses that travel along the cochlear branch, therefore the sense of hearing. Okay, so equilibrium is a vestibule and semicircular canal function. hearing, audition, is a cochlear function. You guys, I'm sure, would like to see that as well. So now I can see it. Cochlear branch emerging from the rear of the cochlea, vestibular branch emerging from the vestibule. And so they're about to merge to form the vestibulocochlear nerve. So impulses for equilibrium, impulses for hearing here. So there are two inner ear senses, hearing and equilibrium, audition and equilibrium. Now, not quite done, taking the cochlea apart, we can reveal, assuming I can actually get it to come apart. Come on, there we go. Looking inside, so you guys can see inside there. Yeah, see inside there. You can see repeated spaces. A green, a blue, a pink space go together, then another green, blue, pink space goes together, then another green, blue, pink, etc., repeated multiple times. And put it this way, the blue spaces contain endolymph. The other spaces, both the green and the pink, contain perilymph. So the blue spaces, the smallest of the spaces, they go by two names. Each blue space is known either as the cochlear duct or the scala media. Either one of those is acceptable names for those. The green spaces, they're known as the scala tympani. Scala tympani space containing perilymph. The pink spaces are known as the scala vestibuli, also containing perilymph. So the blue spaces, scala media or cochlear duct, contain endolymph. So you see that repeated multiple times throughout here. And you can also see the cochlear branch of cranial nerve 8 emerging from the rear of the cochlea. Any questions about what you're seeing in that view? Okay, put this back this way, so I'm going to leave this off because this particular model is also inner ear, specifically cochlea, and even more specifically, just one set of three spaces. In other words, this represents one green, one blue, one pink space here. So remember, the smallest of the three spaces is the cochlear duct or scala media, and it contains endolymph. Then you've got the other two spaces. This structure right there is called the vestibular membrane. If you remember that, it should be fairly easy then to associate it with the scala vestibuli. The scala vestibuli goes with the vestibular membrane here. So this then, by process of elimination, has to be the scala tympani. The space is fluid-filled, perilymph-filled. Scala tympani, scala vestibuli, also a perilymph-filled space, separating the scala vestibuli from the scala media. The cochlear duct is the vestibular membrane, and the small space contains endolymph. And within the scala media / cochlear duct, we've got something called an organ of Corti right here, or spiral organ. It goes by either name, organ of Corti or spiral organ. And it consists of a group of hair cells. You can see hair cell ending with hairs, hair cell ending with hairs, hair cell ending with hairs, hair cell ending with hairs. The hairs are actually what are called stereocilia. So these cells, these hair cells, are not neurons, but they act like neurons. They can actually depolarize and secrete a neurotransmitter. So for all intents and purposes, they're acting very much like neurons here. And so the stereocilia, the hairs of the hair cells, are acting like dendrites. They're the sensory ends of each hair cell. Okay? So the hair cells rest on a what's called this white layer here, called the basilar membrane of the organ of Corti. And then there's a roof over, the roof portion of the organ of Corti is called the tectorial membrane. Tectorial actually does translate as roof there. So the roof membrane, the base or floor membrane, so basal membrane, tectorial membrane, hair cells with stereocilia. All right. In fact, what actually happens is when the endolymph, when sound waves hit the tympanic membrane, it causes the fluid inside the inner ear to begin to move back and forth. What happens is, as the endolymph moves one way, the stereocilia have ion channels that close. When the fluid moves the other way, the stereocilia bend the other way, those ion channels open. And when they open, you get depolarization. And so the hair cells secrete a neurotransmitter called glutamate onto, see these yellow, these are actual neurons here, axon, cell body, axon, actually these are unipolar neurons. They look like they might be bipolar from the way they're imprinted on the model, but there's actually afferent sensory pseudo-unipolar neurons, axons, cell bodies, axons there. So the neurotransmitter, glutamate, from the hair cells is excitatory to these pseudo-unipolar neurons. So they depolarize the threshold, fire an action potential along cochlear branch axons. So these axons here, right there, these cochlear branch axons. You can also see, look at the merger there. So these are vestibular branch axons coming in from the vestibule, and here they are merging. So right here is cranial nerve 8, the vestibular cochlear nerve. Oh, and one last item. What bone is this?
Temple.
Temporal bone. There you go. That's everything I can think of to say about these two models.
Okay.